So HBV cure development has been fantastic with many approved treatments, tenofovir, entecavir, TAF, and now we have maintenance therapy, and patients with Hep B do not make events like decompensation, cancer, so they have a good life. But for Hep D, we need a cure. HDV might be underestimated. You have seen George and Mario, and maybe since we don't test, maybe there might be more with 60 million. There has been recent meta-analysis showing that maybe we have more patients. We don't know the exact number. Are we under or overestimated? Delta hepatitis increase the risk of cirrhosis. HDV requires only a very small amount of HBS to complete viral packaging. We have no available therapy except Peginterferon with many failures and many safety concerns. So we need an HDV cure because we have many patients going to cancer, complication, and death. The virus, you have all this information in the paper, will be published in Liver International. It's a small virus, 36 nanometers in diameter. It needs the B replication because it's the same viral envelope with HBS. There's a small HDV RNA genome, and we have also the HDV ribonucleocapsid. So it's a very interesting, and we have to learn to understand the viral cycle to develop targets and new drugs therapy. The life cycle of HDV and targets for new drugs in development. And I want to thank also Dimitri who helped me for these slides. You see the HDV have the entry through the NTCP receptors. And just to show you the drugs in development and the mode of actions, you know that entry inhibitors, bulivertid, will block the entry. So it has interest in delta and of course also in HBV drug developments. Then the genome go into the nucleus. You have several steps for viral replication. I will not go in details, but we can have discussion during the coffee break. And then you have also the prenylation, and lonafarnib inhibits HDV prenylation through, which is very important for packaging and secretion. So during the next part of the lecture, you will discuss on entry inhibitors, bulivertid, and prenylation inhibitor, lonafarnib, who are in advanced drug development. But we have also HBsAg secretion inhibitors in development. So introduction, we need HDV cure, and we need to improve knowledge on virology, on targets to develop the drugs. So first, entry inhibitors, bulivertid. Bulivertid specifically binds to sodium thoracolate co-transporting polypeptide, NTCP, at the basolateral membrane of differentiated hepatocytes. It has a strong inhibitory effect to HDV infection, and it exclusively targets parenchymal liver cells. So you know that NTCP has been identified by the Chinese group as the receptor for B and delta. Bulivertid has been dosed to more than 600 patients with delta, B, and healthy subjects. Monotherapy-induced HDV RNA declines and improve ALT levels in patient delta in the MIR 202 trial. It has been presented by Heiner Wedemeyer at EASL, and also the combination of 2 milligrams or 5 milligrams with Peginterferon induced synergistic effects. And it has been designed in US and Europe orphan drugs with FDA breakthrough and European Prime. You see it was presented last WSLD meeting by Heiner, the MIR 203 extension study design with 10 milligram bulivertid, so high dose plus Peginterferon, in comparison with bulivertid plus TDF. And you can see the results with a strong antiviral effect, reduction in HDV RNA, and you can see median HDV RNA log reduction in the combination of bulivertid plus Peginterferon, which was a synergistic effect. You see the individual HDV RNA kinetics to see for all patients and not only the median. And you can see that all patients in both treatment arms achieve a more than 1 log decrease in RNA. The HBsAg response, more than 1 log decline or undetectable. You can see that for the arms of 2 milligram plus PEG or 5 milligram, there was more than 1 log decline in 46%, 20%, few in the 10 milligrams dose, and none in the Peginterferon arm. There is ongoing clinical trials. The MIR 204 included 175 patients in combination with PEG. Enrollment has been completed in last December. Patients are randomized in 4 treatment arms. You have the standard of care, Peginterferon. You have 2 milligrams plus interferon, 10 milligrams plus interferon, and you have a mono arm with 10 milligrams bulevirtide. The patients are randomized. The primary endpoint is sustained virological response, defined as negative PCR results for HDV RNA at week 24 after end of treatment. And I agree with Pietro that we should change maybe the definition, the sustained virological response. It's not similar to Hep C. We have to have long-term follow-up to follow the patient. But I think that if we have also this primary endpoint, it's encouraging. We need also long-term endpoint as no events for decompensation, for cancer, and I think this will follow. This study is ongoing in France, in Russia, in Romania, in Moldavia, and many French investigators are among the attendees. There's also other ongoing clinical trials, MIR 301, with 150 patients, 3 treatment arms in 6 countries, Germany, Russia, USA, Georgia, Sweden, Italy. And you can see the different arms with different doses of bulevirtide, 2 and 10 milligrams. Also in France, there's an ATU, it's access treatments for patients. It's bulevirtide 2 milligrams, self-administered by the patients once daily. It's more than 90 patients have already been included, and it's ongoing. Any doctors, hepatologists can ask for the French health authority for the ATU for his patients. The inclusion criteria are compensated liver cirrhosis or severe fibrosis grade 3, evaluated by either liver biopsy or fibroscan, or fibrosis grade 2 with persistent ALT elevation. Exclusion criteria is decompensated liver disease, because at this time we don't have enough data for decompensated liver disease. So also I encourage, if you need this, it's very simple, because you have just 1 page to file for information to send to the health French authorities. Finally, summary for bulevirtide. It's an entry inhibitor, so it binds to NTCP, high efficacy and favorable tolerability. High dose therapy, 10 milligrams, was safe and well tolerated. Bulevirtide monotherapy with 10 milligrams, continuous linear DNA RNA decline over 48 weeks, rapid ALT reduction normalization, and 5 milligram BID was comparable to 10 milligrams QID. And in the combination with PEG, 87% of patients become HDV RNA undetectable at week 48 of therapy, which could be also considered for patients with cirrhosis, bridging fibrosis, high activity inflammation, high ALT, a maintenance therapy, a therapy that blocks the inflammation, blocks the virus, finally allows the RNA to be undetectable, the normalized transaminases. Let's move to prenylation inhibitors, lonafermib. Lonafermib is a small molecule, first-in-class, oral prenylation inhibitor. It's well characterized in patients because in several indications, there has been a large number of patients who have been treated. 2,000 in oncology program by Turing Merck, more than 90 children, those in the Progeria program by Boston Children's Hospitals, more than 170 patients, those in the Delta program. And with longest duration of dosing with more than 10 years, in particular also in children with progeria. Most commonly reported adverse events are GI-related, class effect, well managed with prophylactic treatments, with antivirals and antiemetics. And it has been also designed as US and European orphan, FDA breakthrough, MEA prime. You can see the phase 2 data, and 2 lonafermib-based regimens have been identified for registration. One all-oral, lonafermib 50 milligram BID, plus ritonavir, boosted by ritonavir, and lonafermib plus Peginterferon. And you can see it's a synergistic effect when compared to the Peginterferon plus tenofovir as a historical group, the HIDIT-1 study. You see the phase 2 program, dose combination and endpoints for the all-oral lonafermib boost with ritonavir. 33% of patients has a more than 2 log decline from baseline viral at week 24. for the RNA, and 47% has normalization of ALT. And if you take a composite endpoint, taking transaminase and decline, 29% of patients achieve this. In the combination of lonafarnib plus peginterferon, 78% patients have a 2-log decline, more than 2-log decline, and 88% have normalization of ALT. This is encouraging data, and it was the basis for the design of the phase 3 data. We have to recognize that it's early endpoint and it's small number of patients, but we have now a very large study with 400 patients that deliver phase 3 study. In this study, with 400 patients, you have 4 arms, with a run-in with NEX, then all oral, lonafarnib-ritonavir, combo with peginterferon, a comparator arm with pegylated interferon, monotherapy as a standard of care, and the placebo, and the 24 weeks follow-up. And this large phase 3 study, primary endpoint at week 48 will be more than 2-log decline and normalization on ALT. Secondary endpoints are very important, histologic improvement, more than 2 points in improvement in inflammatory score, no progression in fibrosis, improvement of fibrosis. The left study, you have seen the interesting presentation by Pietro with lambda interferon, who could have a better safety profile. Lonafarnib plus peginterferon for delta, and you can see early result that was very interesting in terms of synergistic efficacy. The summary for lonafarnib, we have high efficacy in phase 2 study, favorable tolerability. Most common adverse events are GI, class effect. There's a synergistic effect with peginterferon. There is an ongoing phase 3 deliver global study, and peginterferon lambda plus lonafarnib combination is in development. There has been regulatory designation as an orphan disease, FDA breakthrough EMEA prime, and we are waiting for the interesting result of the next study. Finally, dear colleagues, ladies and gentlemen, the take-home message. HDV is a defective virus which needs HBV replication present for its own viral replication. HDV, similarly to HBV, infects hepatocyte via high specific interaction with human NTCP expressed on basolateral membrane of hepatocyte. Current treatment of care is peginterferon for 48 weeks. For patients who fail interferon, there is no therapeutic option available. Drugs in development include entry inhibitors, mercludex, prenylation inhibitors, lonafarnib, and HBsAg release inhibitors. For bulivertide and lonafarnib, the addition of peginterferon appears to be synergistic. There is a need for long-term follow-up of patients, long-term endpoint, strong and robust endpoint, as no events, no cancer, and no decompensation. HDV cure program will also lead to HDV cure, but in a long term, so we need a rapid HDV cure program. We need this HDV cure program. I think that I have convinced you. Thank you for your presence.